Here we will explore true facts about meiosis. At first glance, meiosis may be nothing more than a weird word that someone attempted to come up in a game of Scrabble. But in fact, it is the process that creates your sex cells. Males produce sperm cells, which fertilizes the eggs found in females and eventually produces newborn babies. These are known as gametes and are found in the gonads of a human body. There are two types of meiosis that are conveniently named meiosis 1 and meiosis 2, because scientific words can't get any easier than that. The cell begins with interphase, and as the cell grows larger, the DNA inside the nucleus replicates and condenses into X-shaped structures called chromosomes. They then attach to its homologous or same counterparts and exchange corresponding DNA segments in a process called crossing over, and this allows the end result to have genetic diversity. To understand this, imagine your joints were detachable and you decide to give your clone your right hand in exchange for his left foot. That is how crossing over works. Once the homologous pairs of chromosomes exchange parts, the nuclear envelope dissipates and thin spindle fibers merge from structures called centrosomes.
Chromosomes line up in the middle of the cell known as the metaphase plate. These spindle fibers attach to the chromosomes and pull them on each side of the cell through cytokinesis. This makes the cell look like a hot pocket that hasn't been fully separated, or possibly an egg of silly putty when pulled apart, or even chewed up bubblegum that has stuck to the bottom of your sneaker. Furthermore, cytokinesis leaves a cleavage furrow which divides the cell into two. Meiosis 2 does almost the exact same thing as meiosis 1, but less confusing. This time, interphase doesn't take place and there's no cell replication. Rather, it continues from the two haploid cells from meiosis 1. There is also no crossing over once the DNA condenses into chromosomes.
The chromosomes still, however, line up on the metaphase plate and are pulled apart and divided through cytokinesis. As said before, crossing over in Gregor Mendel's law of independent assortment allows genetic diversity within the cell. DNA from homologous chromosomes cross over to each other at random spots on the chromosomes and can occur at multiple times. The chromosomes are now unique and different from both the mother and father's DNA. In independent assortment, chromosomes are randomly paired. For humans, there are 8,388,608 combinations. This factor in humans shows the large possibility for different combinations of chromosomes. This is an animation of another cell dividing process called mitosis. As you can see, meiosis looks virtually indistinguishable from mitosis. They perform almost the same actions in replicating cells. However, mitosis creates two identical somatic cells that are termed diploid because they contain 46 chromosomes with two sets of genes. Meanwhile, meiosis creates four genetically diverse gametes that are termed haploid because they contain only 23 chromosomes and one set of genes.
And, like all future robots, the process of meiosis can malfunction and have errors. These errors can be either harmless, or they can have profound effects on the genes of the daughter cells. A non-disjunction occurs when sister chromatids or chromosomes fail to separate. Another error would be when homologous chromosomes misalign with each other. This means that when a piece of their DNA is exchanged, there are deletions or duplications. Errors in DNA replication can affect whether genes in the daughter cells work properly or whether they are mutated. Furthermore, errors in the process may cause disorders and other mutations such as Down syndrome, which is when the cell has an extra copy of chromosome 21.
Just remember, if someone ever hurts your feelings, imagine if you were the first sperm to ever reach the egg.